So I'm going to talk about some work I've been doing. This is work very much in progress. It's work with an experimental colleague of mine who's at the Stem Cell Institute that's next door to my institute, Sunil. And he's a yeast biologist, and what he does is he grows yeast in these chemostats, these sort of volumes. It allows him to do is to grow these yeast cells in a volume that's well mixed, well stirred, and yet open. So he's able to put in nutrients, take out waste, and so he's able to sort of set up roughly kinds of like steady state conditions. So for instance, he can set up this chemostat so that the number of cells are constant. And in that kind of a condition, he's able to sort of ask questions about how these yeast are growing, what kind of behavior they're displaying, and so on. So one of the really interesting behaviors that's been well known for a long time is that if you grow these cells, if you starve them in pretty close to starving conditions, you give them a little bit of food, but they're not growing very happily, they are a little stressed out, then you can get these beautiful oscillations. So what's plotted here is the dissolved oxygen. And the dissolved oxygen is a sort of a proxy for cells that are doing respiration, which is something they do when they grow and divide. So what's happening over here is that there is a portion of time where cells are not growing and dividing. Then suddenly, in a synchronized fashion, some cells go into this burst of growth and cell division. Then it gets quiet again, and so on. And remember, this is a well-mixed system, and this is millions, and in fact, more than millions of cells, hundreds of millions of cells. So there is this switch from the quiet phase to the growth and cell division is really a lot of cells sort of synchronously moving into this stage. And people have managed to keep this oscillation running for like weeks, almost a month, like 3 to 4 weeks, which is hundreds of, or at least 100 cycles. So one of the things you'd notice is, if you look at this, is that there seem to be at least two time scales. There are these fast transitions from here to there, and maybe much slower stuff happening in these phases. So, that might remind you, as it did me, of certain kinds of oscillations we see in physical systems, which have two time scales, such as Van der Pol oscillators and so on. And one of the things I wanted to do is to ask whether you can understand this kind of yeast oscillation as a relaxation oscillator. But to tell you a little more about some of the biological experiments that have been done, people, including my colleague Sunil, have looked a lot at the state of the cells in these different phases of the oscillation. They've looked at gene expression data, and they can really see sort of, broadly speaking, two very different metabolic states. One is a sort of a growth and cell division state where there are all the genes that are involved in growing, replicate DNA, things like that are on. And another state which is very reminiscent to the kinds of quiet states that are seen also in other kinds of cells, in human cells and mice cells and so on, which biologists are very interested in because they are involved. This kind of switch between quiescence and growth is something that's involved a lot in cancer, for instance. And also, for instance, in stem cells, when you, for example, cut yourself and the wound is healing, it starts by stem cells going from a quiet state to a growing and proliferating state. Also in development, all over development of an embryo, there are these constant switches from growth and proliferation to quiet states to back to growing states and so on. So this kind of question has been of a lot of interest for biologists. And given all the technology in the last couple of decades, people have studied this at the single cell level to a great extent, with a lot of gene expression data and looking at the gene networks and proteins. But what's of, I think, interest with this yeast system is it forces you to sort of focus back on the fact that a lot of these phenomena in cancer and tumors or in tissues are happening in a population. So a lot of this is really collective behavior. In the case of these oscillations, it's rather obvious. It's this synchronized jump into a growing, proliferating state. And that's something this system sort of forces you to confront again. Another thing this yeast system kind of forces us to bring the focus back on is the metabolism and the metabolic state. A lot of work has been done on gene networks, proteins, what genes are happening and changing in these switches of states. But what's happening to the small molecules, the ones that are providing energy, or carbon atoms, or nitrogen atoms? And that's, again, something that in the yeast cells is much easier to access. It's also more obvious to sort of know what the state is, as opposed to other kinds of systems that biologists have studied. So what I wanted to do, I should have mentioned that also in various experiments with yeast, these two broad states of growth and cell division have been sort of seen in situations where it's the same environment, and yet the cells can be in these two states. So there is a hint of an underlying bistability here somewhere. So people don't really know exactly what's causing this bistability. But what I wanted to do is ask whether the oscillations can be sort of modeled as a sort of a relaxation oscillator, but one that has an underlying bistability. Can one take an underlying two-state system and sort of make an oscillator out of it? So it turns out that there is a neat way of doing that for any kind of bistable system or a large class of bistable systems. So for instance, suppose you have it's even a one-dimensional ordinary differential equation, which exhibits bistability to two stable states for a certain range of a control parameter, flanked by monostable regions. If you have a system like this, under very broad conditions, by adding one link and making this control parameter itself a dynamical variable and adding a suitable link, you can actually make this into an oscillator very easily. And the idea is the following. Suppose this is a very slow time scale interaction. So on a fast time scale, the system will quickly jump to one of these branches of the stable states, and then it'll start creeping on a much slower time scale along this. So if A is large and it's sort of inhibiting this alpha, it'll start creeping down until it falls off the edge, jumps to the lower branch, starts creeping up, and then jumps back up, and so on. So this kind of addition of a link sort of frustrates, in some sense, the underlying bistable system, and you get this sort of two-time scale oscillator. Okay? So we wanted to take, there's a lot more mathematically precise statements one can make about this, but I'll leave that for you to read up on or ask me later. But I just wanted to use this idea of this frustrated bistability in the context of these yeast oscillations. So what we do is we keep track of the number of quiet cells, the number of growing cells, and a resource that's present in this system, and put in various kinds of processes that we know are happening. For instance, switching between these two states, growth and dilution, feeding a new resource into the system, which is what Sunil does, consumption of the resource, and so on. And one can make this kind of frustrated bistable system. And I probably won't go into details because there's not enough time, but you can start, once you have a model like this, you can get oscillations that resemble the yeast oscillations, and one can do the usual things. One can ask, can one make predictions and go back to experiments and see that you're getting the same kinds of things happening and so on. And one can ask, what does this kind of model imply for the underlying biological system? So one of the things, for instance, just as an example, one can say is, if these switching rates are constant, and that is switching is random, you'll never get oscillations. We can prove that. What you need is for them to be functions of and non-linear functions, typically, of the fraction of quiescent cells, for example. So one can interpret that as saying that, well, the cells need to know what the density is, which means they need to be able to communicate somehow. And that opens up a whole set of questions for the biologists. They can sort of go and ask, how is this communication happening? Is it a diffusible molecule? Is it by cell-cell contacts, and so on? One can also do, other kinds of basic predictions, which I again won't go into because of lack of time, but things like what happens as you change parameters? Do you see in the experiments that the oscillations break down at the right point, and so on? And we are seeing that, and we see a pretty good match with experiments. But let me focus on in the 3 minutes I have left, on this question that I think is the most interesting direction to take, of what is the metabolic state and what is the metabolic drivers of this switch between growth and quiescence. So in the model, we have up here sort of the oscillation, and we have this resource that I was talking about. Now you can think of that as an external resource, which is in the environment, but it could equally, in such an abstract, simple model, could also be an internal resource where you think of this model as being some kind of mean field-like model where we're assuming every cell has the same average internal resource. Whatever you interpret this resource to be, you can sort of see in the model the kind of trace it follows. So what Sunil did was he took about 100 different metabolites, and by taking samples along these different stages of oscillations from these yeast cells, measured the level of different metabolites in these yeast cells. And he found 2, and only 2 out of all of these that he looked at, which sort of had a reasonably similar sort of profile. Many of the others don't even oscillate. Many others that do oscillate have the wrong phase. They go up at the wrong time and come down at the wrong time. But these two, Acetyl-CoA and NADPH, seem to be sort of, reminiscent of this. And if we look a little closely at what these molecules are involved in, what kind of chemical reactions, we think we can make a pretty good calculated guess that it's Acetyl-CoA. This is a pretty flexible and interesting molecule. It's involved in all sorts of things, from the TCA cycle, which is the thing that sort of produces energy. It's the energy source of a cell to some extent. But it can also be involved directly in production of stuff that you need as you're growing and dividing. And it can also regulate the genes indirectly that are involved in turning on or turning off other processes that are involved in growth. So people don't know and have no real evidence for how this is regulated in detail in these kind of switches between growth and cell division. But one can imagine that it's plausible that in one scenario, and there are people who have sort of made this plausibility argument, that if there's very little acetyl-CoA, it sort of gets shunted into the energy producing mode, so that it's producing just the minimum amount of energy you need to carry on and just survive. And when there's lots of it, it gets shunted into these other pathways where it's actually helping to produce some new molecules that are required for growth. So people have made this, and this sort of plausibility argument without any real evidence behind it. And I think what we are doing is, with our models and the experiments, adding a little more, just at least one step more evidence and one more way of looking at it. Because one of the things that you can do with the model is sort of ask a little more detailed questions about what this regulation should look like. What are, for instance, the threshold values of acetyl-CoA which cause a switch from there to there? What kinds of effects should one look at on the growth rates or on the switching rates, for instance? Because you can use the model to try and predict which works and which doesn't, which gives oscillations, which doesn't. So those are the kinds of directions we're going in. Experimentally, let me just end by saying Sunil is going to do a lot of experiments which try and do exactly this: manipulate, measure acetyl-CoA, see if it matches what we expect from the model. And at the model end, one of the things that I didn't have time to go into, but is precisely what I was saying, that the model can sort of predict when and where the oscillations will happen. And one of the things that we do predict is specifically the way acetyl-CoA can affect the switching rates is kind of a little counterintuitive. And that's maybe not surprising, because it's a very, very simple model. And it's such a crude model that, for instance, one of the things we don't have is the fact that different cells may have different amounts of this internal resource. In fact, it's very plausible that cells in different states will have different amounts of resource, but even cells in the same state, if it's quiescent or growing, will also have different amounts. So that's one direction that I'm actually going in, and I have constructed a model where your cells are allowed to have different amounts. And that's a place where some of these counterintuitive elements can start to get resolved. So, maybe next time in the next meeting, I hope to have more details and a little more of a coherent story. But I think we've taken a couple of steps towards trying to understand this oscillation, at least to the extent that we know we have a good guess on what is driving it. So I'll stop here, and thank you for your attention, and I'll take questions.
Any questions from the top?
Sandeep, since you talked about this information, information has to be traveled.
Yes.
Right? Yeah. And we also know that in delayed system, oscillations can also come. How relevant would it be to...
Sorry, in what systems?
Delayed.
Oh, delayed systems. Right? Yeah, sure.
Even one delayed system can show up.
Sure. Yeah, absolutely.
Can one include delay practically and make system even less dimensional and get an even simpler model?
It probably is possible. The question you're asking is there an effective time-delayed 1D differential equation which behaves the same as this two-dimensional equation that I had? Yes and no. I suppose that it's certainly possible to just plug in a delay and get sort of similar behavior. I suspect that is the case. Is it relevant for this? Well, the advantage of the two-dimensional system is that the delay in some sense is not put in by hand, but emerges out of the natural dynamics of the system. In terms of parameters that are more easily experimentally tweakable because it basically, the delay comes from the resource build-up. And the resource build-up is dictated by the rate at which you are adding resource and the rate at which you're sort of diluting it, and which are direct experimental knobs that are there. So in some sense, the two-dimensional system allows you to make a more direct connection with the experiment. But in terms of a more, if one doesn't worry about just this experiment, but is sort of talking about an oscillations in populations in general, maybe a delayed 1D equation allows you to explore a little more broadly the minimum conditions you need to get it. Because there might well be other populations in which it's not, the delay is not coming because of buildup of a resource, but some other threshold effect or something. So maybe it's useful for sort of a little more broader sort of view of these things.
Let's thank the speaker once again.
Thank you.